So in this unit, we're going to learn about monohybrid and dihybrid crosses and how the characteristics are inherited in each of these crosses. You're going to learn more about alleles of genes and which ones are usually dominant or recessive. And we're going to take dominant alleles results in complete dominance in a hybrid. Some alleles are not completely dominant and result in incomplete or partial dominance. And equally dominant alleles will result in codominance. So you'll learn about all of those: completely dominant, partially or incomplete dominance, and codominance. And you'll learn about how these affect the alleles in the hybrid offspring. We're going to learn how to do genetic diagrams so that we can actually solve some genetic problems. Okay, so when we're looking at our homologous chromosomes, what we end up realizing, let me just get to my pen quick sticks. We know that we're looking at alleles. Again, what is an allele? It is just a variation of a specific gene, right? Now, these variations, gene. This pen doesn't want to work with me today. So, these variations can either be homozygous or heterozygous. Now, homozygous, don't get scared, it's just terminology. Homozygous is a cell is said to be homozygous for a particular gene when the identical alleles of the gene are present. So the alleles must be identical and they are present on both the homologous chromosomes. Okay, so if you look here, this is a homozygous allele. They are present on both the homologous chromosomes. Do you see here? We end up having a homozygous. They have the same letters. Both are small or both are big. That is an allele. An allele is a variation of a gene. For instance, this could be your brown hair and that could be your blonde hair. On this chromosome, we end up having two that are exactly the same. And on this one, we end up having two that are exactly the same, but they're the recessive type. Okay, dominant, recessive. Now, heterozygous. This is a diploid organism is heterozygous at a gene locus when cells contain two different alleles. Do you see that? The cells contain two different alleles. One wild type allele and one mutant. Guys, one wild type ends up just being the original allele and then it mutated slightly to allow for the other allele. Okay. The cell or organism is therefore called heterozygote. So here we end up having our wild type allele and our mutated allele. And because they are next to each other and they are different, they are called heterozygous. Different means hetero, same is homo. Homo, homo, hetero, hetero. Okay, homozygous, heterozygous. Very important to understand the difference between the two. So when we look at the genetic makeup of an organism, we've just discussed homozygous and heterozygous. So let's just see if we can identify this. This is two of the same alleles, which means it ends up being a homozygous, right? Zygous. This one is two different alleles, which means it's heterozygous. And this one is two of the same, which means it's homozygous. Zygous. Sorry, my writing is not winning on this platform, but it's okay. We'll keep going. Now, let's talk about genotype and phenotype. We need to understand the definition of genotype and the definition of phenotype before we can even attempt to understand what's going on this page. So, our definition for genotype is the alleles present in the organism. And for phenotype, it's the visible expression of the genotype or the visible expression of the alleles. Okay, so let's go back to genotype. Genotype is the alleles present. It's the genetic code hence genotype. It's the genetic code basically that tells me my alleles. What is my allele that I'm looking at? I'm looking at A and A. I'm looking at capital A and little a, and I'm looking at AA. My genotype for this individual is a homozygous genotype. It's a heterozygous genotype, and it's a homozygous genotype. Okay? When I'm talking about the phenotype of something, what I want you to understand, it's the visible expression. Visible means you can see it. Hey? Let's draw a little eye here with little eyelashes. You can see the phenotype. And what are you looking at? Well, you're looking at what the gene is telling you it is. This gene is telling you we've got a dark butterfly. Okay? Obviously, A, capital A, codes for dark. And because it's a homozygous dominant, this is dominant, these capitals are dominant, right? So it's a dominant gene. We end up having a visual expression of this gene, which shows you that it's dark. Here, we end up having both alleles, both allele types, a dominant and a recessive. So here we've got our dominant, which is our wild type, right? And our recessive, not necessarily always the wild type, but in this case, let's make it the wild type. And this is the mutated allele from that wild type, right? And because this is a dominant gene, in this case, it's a heterozygous, but we see it as being dark, okay? Which is very scary because just because it looks dark doesn't mean we can assume that its genotype is AA. We must also allow for it to be a recessive being masked by a dominant gene. Okay, so those are the two options. Because our phenotype is dark, we then can assume that it's one of those two. We can't assume it's only the one. Over here, I see a light-colored moth. And if, in this case, the A is the dominant and it masks the recessive, I will only see a light moth if it's homozygous recessive. We can automatically assume that its genotype is AA, and it is not a combination of those, because we would only see a dark moth with the dominant genes which marks the recessive gene. The only way that we'll see the light moth is if it is a homozygous recessive genotype. Again, your genotype is showing you what alleles are present in the organism. Your phenotype is your visible expression. Just because your visible expression, in this case, is the same, it doesn't mean it has the same genotype. But we can assume that because there is a dominant allele and it masks the recessive, if we see a light-colored moth, we can then assume that it is homozygous recessive. Recessive in this instance. Okay. Okay, so we're going to look at meiosis and the segregation of our alleles. Now, firstly, we know that when two parents mate and produce an offspring, the offspring receives an allele from each person or each parent, and each allele carries a specific characteristic. So to understand how alleles separate and appear in offspring, we need to look at meiosis again. When reproductive cells undergo meiosis to form sex cells, this is your reproductive cell. This is the whole process of meiosis, meiosis 1 and meiosis 2, to produce our offspring. Remember, it's a 25% chance for each of these to be created. Okay? The diploid parent cell must produce haploid gametes. So this here is a 2N cell because we've got 2 chromosomes They each have a locus that has a specific gene on it. Over here, we can see there's only one chromosome with one allele on it. So this ends up being your haploid cells. Okay. At the end, there will only be one allele of this gene that was originally in the parent cell. There will only be one allele in each of the cells at the end, okay? Because the other one has to fuse with the egg or the sperm, and then two of them, then your cells go from haploid and become diploid again. So, when meiosis takes place during the formation of gametes, the homologous chromosomes separate. Each gamete receives only one allele pair, and if the pair is made up of different alleles, the gametes receive either a dominant or a recessive, or a wild type or a mutated type. Okay, so let's go through this diagram. I'm going to change it to a eraser. I don't know if you can see this, but here we end up having our 2N diploid cell. Before it undergoes meiosis, it has to replicate itself. It has to create two of itself. It duplicates itself, and there we've got our our double homologous chromosomes that have replicated itself. In this process, we undergo meiosis 1. And during meiosis 1, we end up having crossover that happens here at chiasmata. This increases our genetic diversity, and we have random segregation of these guys along the equator during metaphase. Once these move into meiosis 2, each homologous pair is then separated by these spindle fibers. They're pulled apart, they go to each opposite side, and you end up getting one homologous pair in each of these cells. And these are now your daughter cells. This is the beginning of prophase 2. During prophase 2, we end up separating this homologous chromosome, and we take the single strands of these chromosomes to the opposite sides with these spindle fibers. And therefore, one allele gets separated on each side. Okay. These alleles are then in these daughter cells, or these are actually in your gametes now. So these are your possible haploid gametes that can exist with their different alleles in them. Each allele codes for a specific item, and if this guy bonds with dad that has a little y, then, or sperm that has a little y, then you're going to have a dominant and a recessive gene. You're going to have a heterozygous haploid cell, diploid cell, like you have right in the beginning here. Okay, so again, we have a diploid cell. It undergoes meiosis 1. It first has to duplicate itself. And in that, once it's duplicated itself or replicated itself, it undergoes crossing over to ensure a little bit more diversity within its genetics. And we end up having random segregation. We end up splitting these apart, and they end up becoming in two different cells. Segregation allows them to move into their two separate daughter cells. In meiosis 2, they split again, smack down the middle, and your spindle fibers pull each allele to the opposite side. And then chiasmata happens. You end up having, not chiasmata, cytokinesis happens, and your cell separates into its two daughter cells again. And these cells are now your gametes. In this slide, we're going to look at genetic diagrams. Genetic diagrams will deal with two types, a monohybrid and a dihybrid. Dihybrid, you just need to understand to be able to see how the Punnett square comes around. But the monohybrid is what you have to do regularly, okay, especially in a test. Now, a monohybrid cross is a cross between two organisms that involves one trait. This is a yellow pod, a breeding yellow pod, and a breeding green pod, okay? This is homozygous for these two alleles together. They're exactly the same, so it's homozygous, but these two are also homozygous. This over here on the left-hand side is homozygous dominant, zygous dominant, and this on the top here ends up being also homozygous recessive. Now we're going to do many monohybrid crosses now, so we can practice this idea. These can change, these can be heterozygous, but at the end of the day, in this cross, we end up getting a heterozygous, and it's a dominant, dominant. Oh my gosh, I can't. I'm mixing my B's and my D's there for a second. Dominant. Well, heterozygous will mean that that trait will mask the recessive trait anyway. So a monohybrid cross basically is just looking at the fact that we're looking at one trait, one genetic marker that dictates what's going on here. A dihybrid cross is a cross between two organisms involving two traits. So in this instance, we end up having yellow and round, which ends up, so that ends up being yellow, and the small y's tell you that it's going to be green. So the capital Y is for yellow. If you have a lowercase y, your color is going to be a green P. And if you have capital R's, or you have an R, that ends up being round, the shape of your P. And if you have a little r, it's going to be wrinkled. But now, depending on the way it crosses and your dominant and your recessive, if there's masking or not. But what I want you to understand is there's two traits in this instance that is being tested for. You have your color, that's your color, and you have your shape, that's your shape. Your color is represented by the Y's, your shape is represented by the R's. Both of those are coded by different traits, different genes on your chromosome. So we end up having our chromosome, that can be the gene location for your Y, and that can be the gene location for your R's. So they are two different gene traits that are being crossed here. And we'll go into this diagram in detail a little bit later, not now. Right now, all you need to know, monohybrid cross, one trait. This is representing the color for this trait. Over here, dihybrid cross, you're crossing a color and a shape. Color and a shape, two different genetic components. We're looking at monohybrid crosses now, and we're specifically going to look at a complete dominance. Now, a complete dominance occurs when one allele completely dominates another allele when both present in the genotype. So if it exists that both are present, that means we have a heterozygous organism. Yes? So, for instance, brown hair and blonde hair. Both alleles exist, and when the dominant one, so complete dominance occurs when one allele completely dominates another allele. You won't visually see the blonde, you'll only visually see the brown in an individual. Okay. A dominant allele is always expressed in the phenotype. That's what I said here. What you visually see is what you're going to get, and that's what you can assume the dominant allele to be. This dominant allele will always mask the presence of a recessive allele. So here, the brown and the brown, we've masked this blonde, which means we're not going to see it, we're going to see a brown. A recessive allele will only be expressed in a phenotype where both alleles in the genotype are recessive. So I will only be a blonde person if I have 2 baby Bs, right? And that just means that I need to be homozygous recessive. And that's the only way that I will phenotypically present my genotype. Okay, phenotypically means that I will show that I'm a blonde person or I have a blonde allele because both of them are little b's and they're recessive. I'm not being masked by a large B. So when we look at a monohybrid cross, we always have to do these steps, and we're going to write these out a little bit later, but I just want you to see this and let's do some Punnett squares with it. In my parent generation, I ended up producing an F1 generation. My F1 generation is my first generation of offspring, okay? And what Mendel found, remember if you listened to my discussion on that page, he found that all the offspring looked the same. Now, why the hell would they all look the same? And then what he ended up realizing is that when he used that offspring, allowed them to grow up, allowed them to pollinate themselves, in other words, one flowering plant, we've got a male side on the one side and we've got a female side on the other side. This flower, the male pollinated the female. It's all on the same plant, okay? He ended up realizing that there were specific ratios that ended up popping out, specific ratios. And he did tens of thousands of these experiments, right? Okay, so if we look at this F1 generation, for instance, and we're going to draw our Punnett square of mom and dad that ended up mating, or these flowers that ended up pollinating each other. And in this instance, we're speaking about yellow, capital, is a dominant allele. Which means if it bonds with the little one, we're going to still have a yellow seedling. And that's what we end up getting here. And he ended up finding that, oh, the first generation all looked the same. How did they all look the same? Well, we know that in this diploid cell here, this is the one parent and that's the other parent. In this diploid cell, we're only going to use one of their genes, right, to be able to continue. So we end up taking mom or dad, and let's do them in different colors. So we're going to do dad is on top, which ends up being dominant Y. Okay, mom is on the side, which ends up being homozygous recessive. We know that each of those has to spread through this, so we end up going Y, Y. This guy exists here, and he exists here. That guy multiplies out to there. And then this one comes down into these blocks. Capital Y, this one also capital Y, capital Y. The order is not that important within it, but either way we know that this Y masks everything. So we know that every single one of these is exactly the same. Each one of these blocks represents 25%. So our genotype is 100% Y, little y. Let's make it a little y so you can see it. And our phenotype is 100% yellow because the dominant masks that recessive, right? So we're just going to have 100% yellow. This is my genotype ratio. And this is my phenotype ratio. Okay. Now, remember the second generation, what he did is he self-pollinated those plants. The male part of that flower pollinated the female part of that plant, and we ended up getting our F2 generation. So our self-pollination happens, and we have the male part, which is one of these, bonded with, mated with another one, right? And this is what we call a heterozygous cross. On this side, these are supposed to be red. Hey, let's make them red quickly. Okay, so these extend through. That's a capital Y that fits into those ones. This one goes this way, and it ends up being a lowercase y. We take the blue, we get a capital Y and a capital Y, and this one becomes a lowercase y and a lowercase y. And what we notice here, if we look at our genotype ratios, and this is where we're going down here, I just like to draw it out. It's here, it's already there. If you guys can read it and understand, you can think about it. But when I draw it, it just gets you guys to slow it down a little and understand it. Here I end up having capital Y, and it is in a ratio to these two, which is exactly the same, capital Y, lowercase y, and to those two, a little y and a little y. Here we've got one of these is to two of those is to one of those. If we want to do it in a percentage wise, we can then say there's 25% homozygous dominant is to 50% heterozygous is to 25% homozygous recessive. That is your genotype. Sorry, I pushed buttons on the last one, so I just want to go back to where we were. We were just at the F2 generation, where we ended up having our heterozygous cross, which ended up being capital Y, capital Y, little y, because you ended up having a heterozygous male and a heterozygous female. Ended up getting your offspring. That was this, and we spoke about the genotype, and it ended off with me saying that the genotypes ratio is 1 is to 2 is to 1. Okay, that's our genotype, because it ends up looking at what the alleles actually look like. Okay, and then we need to discuss the phenotype, or the phenotypic ratio. And your phenotypic ratio, remember, is what it looks like, its physical characteristic. Because you end up having your dominant alleles in these two, this means that these are going to be yellow peas, or whatever these things are, round balls, and that's going to be yellow. So phenotypically, our ratio is 3 is to 1. Phenotype is 3 is to 1. We're going to have 3 yellow round balls or pods or seeds, in this case little peas, right, is to 1 green. Remember, the green will only be present if it is in a homozygous recessive format. And there we've got 1 homozygous recessive format. These two together gives you your 3. So 75% of the plants that are being reproduced from this organism will be yellow seeds or yellow pods, and 25% of them will be green pods. Okay, so let's look at a couple other complete dominance examples. I'm just going to repeat some in here anyway, but I'm trying to get you to understand what these words mean. So a homozygous dominant, if we're going to look at alleles A and A, for instance. I'm not going to complicate it. All of them are going to be A's, okay? Homozygous dominant. Homozygous means the same. Dominant means capital. Okay. Homozygous recessive ends up being the same but small. So we're going to cross a homozygous dominant organism with a homozygous recessive organism. Pretty much like what we did with that pea plant, okay? Which we already know. You should already know what offspring you're going to have from this, your genotype and your phenotype. I'm going to talk about it every single time. So when you've got 2 homozygous individuals mate with each other, and the one is a dominant and the other is recessive, you're going to get That A comes into both of those. This A goes into there. This capital goes into both of these, and this A comes across into these two. So what do we notice? All of them are exactly the same. So we end up having 100% genotype. And what is that genotype? It is a heterozygous genotype. And we end up getting 100% phenotype. Remember, in a complete dominance, your A, whatever it is going to mask your recessive gene. So all of these are going to look the same. Let's go look at homozygous dominant and heterozygous. Homozygous dominant, guys, A. Heterozygous, A, little a. So we're going to have a dominant with a heterozygous. Bring this A down into both of these blocks. Bring this A down into both of these blocks. And bring this A across. And bring the little a across. Now, genotypically, we're not all the same. But that is exactly the same and that is exactly the same. So genotypically, so our genotype, sorry, I don't know why it does that. Now I can't even spell or write. Hold on. Genotype is equal, our ratio ends up being AA is to AA, which ends up being 1 is to 1, right? I know it's 2 is to 2, but we put it in our simplified version, 1 is to 1, genotype. And our phenotype ends up being what? If I could write properly, sorry, I've been doing these videos all day. Our phenotype ends up being, all of these here have got dominant alleles in them. And therefore, all of this is going to be 100% the same, visibly, right? I don't know what that visual would be if it's A. I mean, it could be brown hair, black hair. I don't know what we've made A, B, okay? But it ends up being 100% phenotype. All of the organisms look the same. Let's go look at heterozygous cross with a homozygous recessive. A heterozygous is that. Homozygous recessive is little a, little a. So, heterozygous, homozygous. This A comes down into these two. This A comes down into these two. This A goes across into those. And this A moves across into these. And what do we notice over here? Also, we end up having, this is exactly the same as that. So genotypically, we have a ratio of heterozygous is to homozygous recessive, which is a 1 is to 1 ratio. And let's try. Phenotype, or phenotypically, we end up having everybody not looking the same. And in this case, we end up getting 50% dominant and 50% recessive. So it becomes a 50% is to 50%, or in this case, a 1 is to 1 ratio. Okay. If you don't understand genotype, phenotype, please, you need to go Google a video now or go send me a message and say that you need an extra lesson on this, because it's very important that you understand this terminology and what it actually means and how we can use Punnett squares to figure out what the genotype and the phenotype will be. Let's do an example. We have a man who is heterozygous for long lashes. He marries a woman who is also heterozygous for long lashes. The allele for long lashes is dominant. And what is the chance that their child will have short eyelashes? So we have mom and dad, so we know we can draw our little Punnett square. we have dad at the top, which is heterozygous, which means he's got a different allele. Let's take L as the allele for lashes, okay? So he's heterozygous, which means he'll have a capital and a lowercase l. And he marries a woman who's also heterozygous. So let's bring her into this, a L and a lowercase l. We apply our Punnett square rules. I'm going to take my L's throughout, and let me bring my male down into this party. What we end up finding out is that we get a 3:1 ratio for our phenotype. Remember, our phenotype is what we're looking for. This is our phenotype. We're looking for short lashes, and short lashes would only be that homozygous recessive, because the capital L masks all of the short lashes in these cases, so that 3 kids out of this one will have long lashes. Okay, so therefore, what is the percentage chance, or what is the chance? We know each block represents 25%. So there is a 25% chance of having a child with short eyelashes. Okay, so this is a type of very easy monohybrid cross, and the examples that go with it. There is this awesome video by Teacher's Pet that I recommend you watch. Every single time she does a question, please pause the video, do the question before she does it, and then see if you get the answer right, and then watch her video. She speaks really fast. It's done really quickly. Watch it a couple times if you don't understand. Message me if you don't understand as well. Okay. In this monohybrid cross, we're going to explore the concept of incomplete or partial dominance. Now, what does that mean? It means that neither allele is dominant. Okay. So we don't have a B, where we've got a dominant allele masking a recessive allele. How do we then write this? We're going to have to understand that we are looking at a trait. So each parent contributes a trait. Let's, for instance, say a color, right? And they have different colors. There's red hair, there's brown hair, and there's blonde hair, for instance. Okay? All of these represent a letter on this color trait. So I can write a redhead as the color red, or I can write it as the color blonde, or I can write it as the color brown. Okay, we'll generally only deal with 2 colors blending because we can only have 2 traits from each parent, right, at a maximum. So this occurs when 2 homozygous parents are crossed and the offspring all look like blends. Now, we've got carnations here in this example. This is the exact same allele as this one, which means it's a homozygous organism. This is exactly the same as that allele, which means it's a homozygous organism. This one just codes genotypically for a white flower, whereas this one codes genotypically to show a phenotype of a red flower. And when they blended that, they ended up finding that they got a red and white mix, which created a pink. And we know this from art. You mix red with white, you're going to get pink. But nature does it too. So let's do this cross. We end up having the one plant gametes at the top, and we end up having the other plant's gametes on the side. If we extend this, the color goes, the white color goes in there, color white, bring the R's down, color R, bring the R's down, color R. What you end up seeing is that all of these look the same. None of them look like their parents, but they all look the same. And these are then a heterozygous organism, right? And what Mendel ended up doing is that he took the parent generation, took the homozygous individuals, blended them, all the offspring looked the same in the F1 generation, then he self-pollinated. So now, what did he do? He went and took a pollen off of this flower and put it onto the female part, the stigma, the style, you know, the stigma, so that it can go into the ovaries and stuff like that on the same flower, on a different flower on the same plant. That makes sense. Okay, so here we end up having our color red and our color white, the gametes of one of these, self-pollinating itself, so its gametes will be exactly the same, and we end up doing our genetic cross. Bring the CR down, bring the CW down, take the CR across, and take the CW across. And what we end up noticing is that we end up having, phenotypically, we would have, this would give me a red flower, this would give me a white flower, but over here I would have pink flowers. So when I'm trying to talk about my genotype and my genotype you end up having CRR is to CWCR is to CWCW, and that is 1 is to 2 is to 1. And what we realize in a partial or incomplete dominance is that our phenotype is going to be the same. We're going to have 1 red is to 2 pink is to 1 white. So here our phenotype and our genotype are the same. If this was a complete dominance, we would end up having red flowers, right? 3 is to 1, red flowers is to 1. But in a partial or incomplete dominance, you have a 1 is to 2 is to 1, and your genotypic ratio is the same as your phenotypic ratio. Another monohybrid cross that we're going to look at is a codominance type of hybrid cross. And blood groups is a very, very cool example to show codominance. The definition for codominance is that 2 alleles of a gene are both expressed in the phenotype, and the individual has the characteristics coded for by both alleles. So this ends up being our blood groups. Now, I don't know if you've ever looked at blood groups, but you get different types of blood groups. You get type A, type B, type AB, and type O. That would be your phenotype. Remember, that is your physical appearance of what your genes are coding for. So that is your blood type. What does a blood type look like? So if I look at blood type A, my gene for my blood group ends up being represented by a letter I, okay? And that I ends up helping me because I now know that on here, this blood group, I end up having these little things, and these little things are called antigens, and you find them on your red blood cells. So these are your red blood cells. Red blood cell O, type O, does not have any antigens on it. But A has a type of antigen on it, B has an antigen B on it, and AB has both antigen ABs, okay? Now, if I have a blood type A, I have an antigen that's sitting on my blood group A, and that means I have an anti-B antibody. If I'm blood group type A, I've got an anti-B antibody, okay? And that floats around in my plasma. So what happens is if I go and I have an accident and they accidentally give me type B blood, these anti-B antibodies are going to attack these red blood cells that have this antigen B on it. And it's going to end up coagulating, it's going to form clumps, and I can actually die from getting the wrong blood type. So I need to get blood from somebody that doesn't have any antigen B on it. So I clearly can't get it from blood group B. I clearly can't get it from blood group AB, because if you are blood group A, if you're blood group B, or if you're blood group AB, you will have antigen A or B or both on it. Okay. Therefore, I will only be able to get blood from obviously somebody that's from my same blood group, a type A, or from a type O, because there's no little antigen B's on this one so that my anti-B bodies attack it. And the same thing goes for my blood group B. Now let's go look at our blood group O, our type O. This does not have any antigens on it, but it has both antibodies, which means blood type O will attack any blood that comes from anybody else. Therefore, type O can only have O-type blood. It will attack all these antigens on the other blood groups. And it is considered the universal donor. Why is it considered the universal donor? Because it doesn't have any antigens on it. These guys are not going to attack this red blood cell because they've got the antibodies in it. Now, if I've got an antigen A, I'll have an antibody B. If I've got an antigen B, I'll have an antibody A. But if I've got an antigen AB, I will not have antibodies A or B in my plasma. So type AB can have any type of blood. They can take blood from anybody because they don't actually have any antibodies that prevents them from taking blood from anybody. And it is therefore known as the universal recipient because it will accept blood from everybody and anybody. Okay. Now, how does this cross work? Well, let's go look. First, the genotype of an A with its antigen A's on it ends up being either homozygous or heterozygous. Okay. Same with B. It has a homozygous trait or it has a heterozygous gene trait. Here we've got AB, which is only a codominance. Here is my codominance. Both of these traits exist and therefore they are going to be present in our phenotype. Okay? And then over here, your little I's end up being your oxygen. We talk about blood. It's oxygen. It ends up being your type O blood. Okay, so let's go do a question and see if we can do a cross. If my mother has blood type B and my dad type A, is it possible for me to have blood type O? Now this is a tricky one. Firstly, we need to be able to look at the different types of bloods. They said that my mother has blood type B. So let's draw. Mom is a B blood type. Now B can be homozygous or it can be heterozygous. Okay? They haven't told us. Dad is type A. So type A can be homozygous A for the blood group, or it can be heterozygous A for the blood group. So I can definitely tell you for certain that if you blend this homozygous with that homozygous, you're never going to get a blood type O. Remember, a blood type O is homozygous like that. So these cannot be their genetic traits that they're crossing. They must be this or that. Okay, this is a huge assumption that we're making. They should tell you in the question a little bit more in detail, but in this case it's fine. We can assume that we're not dealing with the homozygous type gametes or organisms. We're dealing with the heterozygous type organism. So here I'm going to put dad is at the top here, which ends up being AI and little I, and mom is on the side here, AB little I. And if we cross this, we're going to get IA, IB, which is an AB blood group, right? Here you're going to get an IB, I. Here you're going to get an IA, I. And here you're going to get an I. So this child doesn't have to worry. She's not adopted. Let's go look at the blood groups. This is blood group AB. That is the only letters that can be combined together to give you an AB blood group, right? This is a B blood group. This is also a B. Not a B blood group. It's an A blood group or blood type, phenotype. And this ends up being my O. So yes, it is possible for you to have a type O blood group if mom and dad are AB. Now, that means that they both have to be heterozygous. Let's go see what happens if one of them is heterozygous and the other one is homozygous. What do you think is going to happen? Let's keep dad as heterozygous and let's make mom homozygous. And that would work the same if I swapped dad around and I made mom heterozygous. So we sit there and we end up crossing this over. We have, this ends up being IA, IA, IB, IB, little I, and this one is IB and IB. So what do you notice here? When we do this type across, we will definitely see that we have a genotypic ratio of 1:1, and its blood type AB is to blood type B. Can I get an O child from this kind of blend? No, definitely not. And the same will happen if I change this to a homozygous blood group and make this one a heterozygous. You'll end up having a 1:1, A is to AB again. And you guys can try that. Test it out and see if we're right. But we need to understand how to do blood groups. We need to understand that the blend exists. We need to understand the different alleles that code for different things. This codes for blood group B as well as that coding for blood group B. And that's where we get a codominance, where the one item blends nicely with the other one. Okay, and that's also why we're allowed to have our certain blood groups, guys. My B's and my B's are allowed to receive blood from my O's because they have a portion of that in them. If we go back, well, you can see it if you go back. So go look at it, go look at how those I's and the IB's and the exist within the blood groups and why they can accept what they can accept. Okay.